Okay, thank you for the invitation. I remember I gave a talk for this conference about 12 years ago, and it's great to have this opportunity again this year. My laboratory studies developmental and stem cell biology of heart generation and regeneration. And today I'm going to share with you some of our ongoing research focused on cardiomyocyte maturation. As many of you know, PSCs have a tremendous potential in cardiac regenerative medicine, so the field is intensively focused on PSC research. So PSCs can be induced from somatic cells by reprogramming, and they can be differentiated into specific cell types, such as cardiomyocytes. And these cells offer opportunities for disease modeling, drug discovery, toxicity testing, or cell therapy. Despite this tremendous potential, the resulting cardiomyocytes are immature, and this has emerged as a major concern in the field, as cardiomyopathies predominantly manifest in adulthood. In humans, cardiomyocyte maturation takes place over 10 to 20 years, and which involves numerous structural, functional, metabolic changes. If you compare iPS-derived cardiomyocytes with adult CMs, you can clearly see that they are very different cell types. So this table shows the comparison of iPS-LCMs with adult CMs. And if you look at the morphology, iPS-LCMs are circular or polygonal as compared to adult cardiomyocytes are rod-shaped. And if you look at the sarcomere structure, the iPS-LCMs are disarrayed, but these adult CMs showed pretty well organized sarcomeres. T-tubules, which are critical for EC coupling for function, are not absent, pretty much absent in iPS-cell cardiomyocytes. And mitochondria occupy a very small fraction of the cell volume in immature cardiomyocytes as compared to adult cardiomyocytes. They occupy about 40%, 20 to 40% of cell volume. And these immature cardiomyocytes rely on glycolysis as energy and metabolism, but adult cardiomyocytes utilize fatty acid utilize fatty acid for beta oxidation, and nucleation is pretty much iPS-LCMs are mononucleated. And if you look at electrophysiological properties, that upstroke velocity is slower because of the low levels of L-type calcium channels. But resting membrane potential is higher in immature cardiomyocytes because of low levels of potassium channels. And calcium handling is pretty immature. And also, if you look at the force, particularly, these iPS-LCMs generate about nanonewton range of force as compared to micronewton range of force generated by adult CMs. So which is more than 100-fold difference in force generation. And iPS-CMs still can undergo cell division, but that which rarely happens in adult cardiomyocytes. So, as I summarize the differences, there are very different cells, but we do not understand how this maturation process happens. To address this, we started with low-hanging fruits, which we used publicly available data sets from prenatal to postnatal mouse hearts and conducted large-scale meta-analysis. And this work was led by my former fellow, Hideki Wasaki, who's now associate professor at Jichi University in Japan. And the principal component analysis of the data sets show that overall gene expression shifts continually in a unidirectional fashion, which is consistent with the process maturation. And interestingly, the most of the genes continue their expression trends throughout the stages, meaning that once they go up, they just continue to go up. And once they go down, they continue to go down throughout the stages. And we explored how differentially regulated genes at each of the stages are involved in biological functions, and found that nuclear receptors like PPARs and associated transcription factors are gradually activated over time, leading to increased activities of cardiac and metabolic pathways. In contrast, the transcription factors and pathways involved in early heart development and cell cycles are inactivated. So with this data set, we inferred a stage-specific GRNs and developed a maturation prediction program, MESTAT. And this MESTAT indexed the cardiomyocyte status fairly well. So you see here, if you plug in embryonic day 9 mouse cardiomyocytes, the MESTAT classifies to early embryonic stages, and E13 cells to mid embryonic, P0 to neonate, and also P56 myocytes to adult stage. So with this algorithm, we tested how far PSCCMs can advance in culture. And we found that the 10-day cultured PSCCMs are early embryonic. And if you culture 10 more days, they advance to mid to late embryonic stages. However, they remain in mid to neonatal stages even after months of culture. So this show that we also culture more than a year, and they are pretty much stuck at in late embryonic stages. So this show that PSCCMs mature to some extent, but they are eventually arrested around the perinatal stage, suggesting that postnatal gene networks are not activated in cultured PSCCMs. So this led us to hypothesize that perhaps early postnatal environment may provide cues necessary for PSCCMs to further mature. To test this, we took a really bold approach, which is to inject PSC-derived cardiac progenitor cells and myocytes into neonatal hearts and incubate them for a month. And this work was done by my former postdoc, Gansik Cho, who's currently director of a stem cell company, NextCell, in South Korea. So long story short, that was the case. These are PSCCMs cultured in vitro, and if these are introduced into neonatal hearts, they became indistinguishable from adult CMs. And you can appreciate the morphology and calcium transients of iPSC-derived and adult cardiomyocytes. And I'm not showing here, but we're also able to model several adult onset cardiomyopathies as well. So together, this suggests that early postnatal factors and gene networks are crucial for PSCCM maturation. To better understand the postnatal regulation of cardiomyocytes, we analyzed their developmental trajectories and heterogeneity with single-cell RNA sequencing analysis, which has emerged as a powerful tool to study cell fate progression. But it turns out that it's not easy to perform single-cell single-cell RNA sequencing analysis of mature cardiomyocytes because of the large size and rod-shaped morphology. In fact, when isolated with conventional single-cell isolation methods, that myocytes get sheared and damaged, and this causes the abnormally high percentages of mitochondrial transcripts, and meaning that the cytoplasmic RNA leaks out, and this makes the data not really trustworthy. So to address this issue, Sraj has developed a new method to isolate large cardiomyocytes, which uses large particle FACS. We named it LP FACS with a channel size of 500 micrometer. And this work was done by the very talented MD Ph.D. students Raj Khan in the lab. So our new approach really allowed separation of the live and rod-shaped myocytes with a time of flight and optical extinction parameters. And as you can see here, that LVFAX purified adult cardiomyocytes retain their morphologies and with intact sarcomeres. And they show the normal range of mitochondrial transcripts, which is consistent with the bulk RNA sequencing studies. The sorted cells were further validated by single-cell net, which classifies the cell types by comparison to established cell atlases. And they were functionally indistinguishable from unsorted healthy cardiomyocytes as well. So we were fairly confident that this LVFAX can be used to generate high-quality transcriptome data. So with this new method, Sean Murphy, another very talented graduate student in the lab, asked the question how postnatal cardiomyocytes undergo maturation. And with the LVFAX, Sean successfully performed the high-quality single-cell RNA sequencing of the postnatal cardiomyocytes. And our initial analysis with TSNE, the dimensionality reduction algorithm, showed partial segregation, which implies some sort of transcriptomic overlaps between stages. And next, we used Monocle to organize the transcriptome profiles at different stages. And this showed the kind of progressive and unidirectional pattern of cardiomyocytes over the course of maturation. When aligned the postnatal stages to pseudotime, pseudotime is a quantitative measure of a developmental progression, we see that average maturation scores increase with age, but in a kind of a heterogeneous fashion, and consistent with the levels of size, consistent with this, the levels of size heterogeneity were also observed. And using the data set, we predicted the upstream regulators by ingenuity pathways analysis, or IPA, and found that nuclear receptors and transcription cofactors as potential regulators affecting the overall gene expression changes during maturation. So we examined their expression profiles. And in vivo, their levels are increased from embryonic to adult stages, with notable upregulation in postnatal stages. And this incremental expression pattern was also observed in cultured PSCMs, but notably, this PGC1 and PPAR alpha levels remain constantly low. So this suggests that these factors are misregulated in culture and may have an important role during postnatal maturation. So to test this idea, we deleted PGC1 at the neonatal stage using AAV-mediated Cre-LoxP system. And this approach creates mosaic knockout cells, so you can avoid early lethality of the animal, and also allows us to determine the cell autonomous role. And the deletion resulted in a significant decrease in cell size, which is quantified here, and their size is pretty much arrested around the P7 to P14. And this suggests that PGC1 is required for cellular hypertrophy. I'm not showing here, but they were also functionally immature. To determine how the PGC1 mediates hypertrophy and contractility development, we isolated PGC1 knockout cells by LVFAX and conducted single-cell sequencing analysis. And the resulting trajectory showed that PGC1 deficiency MCMs indicated in red becomes less heterogeneous at P7 and maintain the lower maturation scores throughout the stages. So this is consistent with the failure to increase in size and contractility. The gene ontology analysis showed that muscle fiber and cell development is significantly impaired in mutant cells. And since PGC1 and PPAR activity is low in PSCCMs, we tested if increasing their activity affects maturation. So to test this, we treated these PSCCMs with small molecule activators. And indeed, as you can see here, the PGC1 activator treated cardiomyocytes become notably larger than control cardiomyocytes. And this hypertrophic growth was also shown in cells treated with PPAR alpha-specific ligands, WY. So the treatment improved the contractile force as well, measured by traction force microscopy. And this suggests that PGC1 and PPAR has an instructive role in PSCCM maturation. So we were curious how they mediate this effect. And to address this, we performed the chromatin IP sequencing analysis and identified the 148 commonly regulated genes by comparison with RNA sequencing datasets. And we found that several novel target genes with unknown roles in maturation, including hippo signaling effector YAP1 and RNA splicing factor SF32. And YAP1 deletion also reduced the myocyte size and failed to respond to PGC1 and PPAR activation, which is quantified here. So this suggests that YAP1 activity is required for hypertrophy mediated by PGC1 and PPAR signaling. I'm not going to go through all the details, but to identify factors mediating contractility, we performed the siRNA-based high-throughput functional screen with targeting 148 genes regulated by PGC1. And among those targets, knocking down the splicing factor SF32 showed the most significant effects impairing calcium handling and contraction. So this suggests that SF32 is required for contractility development mediated by PGC1 and PPAR signaling. So to summarize, the PGC1 and PPAR signaling is a kind of key regulator of hypertrophy and contractility development during cardiomyocyte maturation. And then YAP1 and SF32 mediate this process. And this shows a cell autonomous and intrinsic mechanism by which postnatal cardiomyocytes undergo maturation. And next, we wondered if there are any extrinsic factors that influence this process. And earlier studies suggested that sympathetic neurons innervate the heart from the epicardial layer to affect the myocyte contractility. So we tested if cardiac neurons regulate cardiomyocyte maturation. In fact, as shown here in red, the heart is an extensively innervated organ, and the idea of neuromodulation of the heart is gaining more attention now because of the importance of autonomic nervous system in heart failure and heart regeneration. However, it was unknown if this can be modeled in PSC system culture. And in collaboration with the lead lab at Johns Hopkins, we successfully generated sympathetic neurons, or SNs. I'm going to call SNs, sympathetic neurons, as SNs from now on, from human iPSCs, and following what's happening in developing embryos. So basically, you can induce the neurocrest cells expressing SOX10, and you can push this more towards sympathetic fate, and eventually you can get this FOX2 positive SNs. So, but when co-cultured with myocytes, we saw that the neurons can be physically coupled to cardiomyocytes, and interestingly, the stimulating sympathetic activity, which was done by nicotine treatment, increased the beating rate and contraction of the coupled cardiomyocytes. So this demonstrates that neurons can control cardiomyocyte function in a human iPS cell model. And based on this, we decided to test the role of the neurons in vivo. And this work was done by my former fellow, Emmanuel Tampakakis, who's now assistant professor at Johns Hopkins. And to do this, we sought to genetically suppress the sympathetic innervation, as the chemical approaches can be non-specific and with indirect effects. And our strategy was to conditionally target the nerve growth factor, or NGF, which is the primary regulator of sympathetic neuron regulation. And we tested several cardiac Cre drivers to reduce NGF levels. And as you can see here, that control hearts showed extensive innervation of SNs and marked by tyrosine hydroxylase. But the innervation is dramatically inhibited when deleting NGF with the SM22 alpha Cre driver expressing Cre in smooth muscle cells. The loss of SNs was also confirmed by reduced heart rate, as shown here. So we use this as an inhibition model. And to figure out what's happening in the heart, we isolated the cardiomyocytes with Langendorf perfusion at P7 and P14, and we found that these mutant cardiomyocytes are smaller in size, but their numbers are increased with higher percentages of phosphohistone 3 positive cells. So this shows that SNs negatively regulate cardiomyocyte proliferation. To better understand how SNs regulate myocyte proliferation, we harvested controlling mutant hearts and examined transcriptomic changes by RNA sequencing analysis. And gene ontology analysis showed increased cell cycle activity, shown in green here, green font, which is consistent with the increased myocyte proliferation. But it was pretty intriguing that circadian rhythm activity, shown in red font here and here, were significantly upregulated. So we further analyzed the genes involved in cell cycle progression and circadian rhythm regulation. And it turns out that the genes involved in mitosis and DNA replication are significantly upregulated, particularly genes controlling mitosis, such as CDK1, CCNP1, and Aurora A kinase. And notably, the genes, the clock genes, among those clock genes, we found that period 1 and 2, or per 1 and 2, were significantly downregulated in mutant hearts as well, which is shown here, RNA level and protein levels. So this led us to test if per 1 and 2 expression is involved in myocyte proliferation. And to test this, we generated per 1 and 2 double knockout mice and examined the effect on the heart. And indeed, the deletion increased the phosphohistone 3 and EDU positive myocytes and reduced the cell size, suggesting that more cells undergoing DNA replication and entering mitosis. And consistently, cell cycle analysis showed that higher percentages of mutant myocytes in the S and M phase And this shows that depletion of the clock genes, PER1 and 2, phenocopies SN inhibition, and suggests that they may be a key mediator of sympathetic signals for cell cycle progression. And this raises a question, if these neurons regulate clock genes, how these neurons regulate clock genes and cell proliferation. So according to previous literatures, adrenergic activation of G-coupler receptors, such as beta receptors, increases cyclic AMP levels, and cyclic AMP response elements are present in the PER1 and 2 promoters. So we decided to test if norepinephrine, which is the main adrenergic neurotransmitter released by SNs, affect the PER1-2 expression. And as you see here, the norepinephrine robustly induced the PER1 and 2 expression, and consistently the treatment suppressed the mitosis genes, shown here as red dots. And however, if PER1 and 2 knockout myocytes are used, shown here in green, the suppression is mostly abolished. So this suggests that this SNs use adrenergic signaling to regulate cell cycle genes, and PER1 and 2 proteins are required for this process. So far, the data suggests that sympathetic neurons regulate PER1 and 2 by adrenergic signals, and now we wondered how they control cell cycle progression of cardiomyocytes. And to figure this out, we analyzed the genes that are known to associate with circadian and cell cycle factors. And among those genes, we found that WEE1 was downregulated, and CDC25 was upregulated in hearts without sympathetic neurons, or PER1 and 2 genes. So what are those factors? So WEE1 is a protein kinase that phosphorylates CDK1 and negatively regulates G2 to mitosis transition, whereas CDC25 is a phosphatase with the opposite effect of WEE1, which promotes this transition. And we found that in the absence of SNs for PER1 and 2, the phosphorylated CDK1 levels are decreased as WEE1 levels are reduced. But unphosphorylated and active form of CDK1 levels are significantly, dramatically increased. And this suggests that PER1 and 2 utilize WEE1 to regulate mitosis entry. So PER genes encode transcriptional regulators, and previous genome-wide studies show that PER proteins are found associated with the WEE1 locus in liver cells. So we performed the chromatin immunoprecipitation using iPSLCMs and found that enrichment of 2 regions within the WEE1 promoter bound by PER2, which is kind of quantified here. And to test this, if the binding sites are functional, we made luciferase reporter constructs without the binding sites and analyzed the activity. And as you can see here, the intact the promoter was able to increase the luciferase activity, but the mutant constructs significantly compromised the activity. So this suggests that the PER proteins may directly regulate WEE1 transcription. So to summarize, our data suggests that sympathetic neurons send adrenergic signals and upregulate clock genes PAR1 and 2, which in turn increases WEE1 kinase levels to phosphorylate CDK1 to negatively regulate the G2 to M phase transition. And we believe that this can be a key extrinsic mechanism to regulate cardiomyocyte maturation. And we are currently investigating if there's any crosstalk between extrinsic and intrinsic signals. Okay, so I'll stop here and happy to discuss and take questions. Thank you.